Good morning, everyone. Thank you all for coming. Welcome to the first seminar series of this season, let's say. Hello to everyone on Zoom. Hi. It's a pleasure for me to introduce Sohini Kar-Narayan, who's in the Materials Science Department at the University of Cambridge. Sohini did her degree and PhD in India, and then she moved as a postdoc to Cambridge, and she hasn't left. She's had a really great career there. She's a fellow of a couple of societies. She's had an ERC starting grant and now has a consolidated grant. In 2023, she was awarded the Peter Day Prize from the Royal Society of Chemistry, even though she's a physicist by training. So you can tell that she's a really interdisciplinary in her outlook. I think from her CV that went around, you'll have seen all the different areas she works in. And I mean, for us, I think it's nice because it shows how science can be so broad across several disciplines, even though she's a physicist and she gets a chemistry prize and she does AFM as well. And she's got a prize for AFM as well. So all of those things, I think she's a very appropriate person to start our seminar series. And I'm delighted you accepted the invitation to come. And we really look forward to your talk, Sohini.
Lona, this is my third time here, and it's always been a pleasure visiting the city. Especially, I was just telling David, it's completely rained out in England, so my family is really jealous, and my group members very jealous when I send them a snapshot of what it's like out here. So really pleased to be here. I've already met a couple of people. David mentioned about my Peter Day Prize, and I just came across someone who was Peter Day's student, which is amazing. Yes, Marta, exactly, which is amazing. Yeah. So thank you ever so much for having me here. I'm going to talk about functional polymers for energy sensing and biomedical applications. I always like to start by kind of giving you an idea of what the scientific focus is in my group. And I like to start with this question that I usually throw out to my first year material science undergraduate students, and that is, how strong is a bar of copper? So the way I do it is, so I have a class of 300 students, and I have a few of these copper bars. They all look identical. And I look around and I throw the questions like, so how strong do you think this is? I look for the most athletic looking person I can find in the audience and say, do you want to give this a shot? And I give them one of these copper bars, and I have one for myself. And I say, go on, bend it. And this person tries really hard, and they're just not able to, just a slender bar, and they're just not able to do it. And I was like, oh, it looks like you're having some trouble. Here, let me show you. And I bend mine, and it's super easy. And then everyone suddenly is like, ooh, what's going on? Because on the face of it, they look identical. But what is different, of course, is their microstructure. So if you look closely, if you look sort of, if you look what's inside, these copper bars are actually made up of lots of little tiny copper grains, crystallites of copper. And depending on the size, the number of grains and the number of grain boundaries, the strength of these copper bars can be very different. So in fact, the one that I had was actually an annealed copper bar. So the grains were slightly larger, fewer grain boundaries. So dislocations could move more freely, which is why I could bend it. Whereas the poor person next to me was left with one which had loads of grain boundaries, which made the material much stronger. And what is the difference really? Well, it's exactly what I said. I annealed my piece of copper, right? So there was something about the processing that changed the microstructure, which then gave rise to these properties that were so different. So that's been the scientific focus of what we do in my group, and that is trying to understand this relationship between structure and properties and using processing as a way of manipulating structures such that we can get the kind of properties that we like. And I showed you a simple example with copper, but then you can extend this to all sorts of materials. In particular, I'm interested in functional materials. When I say functional, it's quite a catch-all term, really. I'm interested in materials that are responsive, where you can apply some sort of, whether temperature, field, whatever, and see some response. Often the response that I'm interested in is electrical response, because I like measuring things. As David mentioned, I'm a physicist by training. I started off by measuring samples, either in the using STM or good old transport measurements. And so I'm always interested in the electrical response that these materials might have. So again, broadly speaking, the materials that I'm interested have applications in energy, sensing, and of late, a lot of biomedical applications. What's common, of course, is the materials. We work with, as I mentioned, functional materials. And we often work at the nanoscale because this is where we can use our processing routes to manipulate the structure. But of course, we need to understand what the properties are at the nanoscale. So we do quite a lot of nanoscale characterization, so a lot of AFM or scanning probe microscopy-based techniques. And at the end of the day, I'm interested in making devices. So there's the materials, but then how do we make them into devices? And so for that, I use additive manufacturing methods. So a lot of printing methods, so not just 3D printing, but more advanced microscale additive manufacturing techniques. Some of them I'll talk about today. So that's sort of where the device engineering comes into play. Just to give you just a brief glance at some of the devices that we work with. So in the field of energy, I do quite a lot of work on mechanical energy harvesting. I'll talk about this in today's lecture. Also some work with thermal energy harvesting. You'll notice that a lot of these are flexible and stretchable because some of the applications I'm interested in are either in wearables or areas where rigid devices wouldn't quite work. In terms of sensing, again, I'll touch on some of these. We've been doing quite a lot of work on microfluidic devices of late, also trying to see how functional materials can interface with biology. So using, for instance, here, piezoelectric materials to study cells. Some work on textiles, making them smart, making them responsive. I'll touch on that today, all the way up to wearable sensors. So these are some very recent work from my group where we're actually looking at analytes or electrolytes or metabolites directly in sweat using these sort of wearable microfluidic devices. And onto biomedical devices, we have gone all the way to sensors for orthopedic implants to soft actuators for robotic applications. Again, I'll just touch briefly on these at the very end. Like I said, what's common to all of these really is the fact that one, we use scanning probe microscopy to understand properties at the nanoscale, and additive manufacturing to go from nanomaterials to devices. So with that, the bulk of today's talk, I'm going to focus on applications mostly in energy and sensing. In my group, we're mostly interested in how can we convert mechanical energy and thermal energy in the environment into electrical energy. And just to give you an idea, the focus or the end goal is to be able to power wireless sensors, so sort of like the Internet of Things kind of a scenario where you have loads of sensors in the environment. They could be embedded in the walls or in infrastructure, or they could be implanted within your body. Normally, how would you power such sensors? You could use batteries, but then batteries need to be recharged or replaced. So there are limitations there. One way is to harvest energy from the environment to power these. And so mechanical and thermal to me are ubiquitous. I often, again, ask this question to my students, like what are ambient energy sources you can think of? And everyone says the sun, which is absolutely right. But for a lot of these applications, and also if you live in England, it's not always the most accessible or convenient option. So anyway, so to go from mechanical to electrical energy, I make use of piezoelectric or triboelectric materials. And for thermal to electrical, I make use of thermoelectric materials. Today, I'll mostly focus on piezoelectric and triboelectric materials. And again, I don't need to labor on this too much, but the idea is that the devices that I'm looking to power are low power devices. So we're talking about sort of microwatts to milliwatts range. And that is feasible from the sort of energy that is available in ambient vibrations, which is not actually that high. And the most convenient way to go from mechanical energy in the ambient environment to electrical energy, at least for me, is to use piezoelectric materials. So if you're not familiar with piezoelectric materials, these are materials which, by virtue of their crystal structure, can produce electricity when they are when you apply a stress or strain. So basically, if you squeeze the material or you strain it somehow, there is a change in polarization, which results in an electric potential developing across the material, and you can have an external circuit and cause a current to flow. The reverse occurs as well. You can apply a voltage across these crystals or these materials, and they will change shape. And you see lots of examples in everything from ultrasound machines to to gas lighters, where we make use of this effect. And typically, these materials are ceramic in nature. So most piezoelectrics you come across, maybe barium titanate, lead zirconium titanate. So lots of oxides which show this effect. And I guess the important thing to note is, you need some sort of non-centrosymmetric, you need a non-centrosymmetric crystal structure for this to work. You need to have a dipole somewhere present in your structure, which when you then, try to move apart or squeeze together, will change the polarization, which is then what produces this voltage across the material. So the existence of a dipole is very necessary. But from a mechanical energy harvesting point of view, these ceramics are not necessarily the ideal candidate, because as you can imagine, I'm talking about something which is, being squeezed or strained repeatedly over and over again. And ceramics are, stiff. Mechanically, they're prone to failure. So this is where I came across another class of piezoelectrics, namely piezoelectric polymers, which also exhibit this effect, but usually, well, not usually, they are generally much poorer counterparts to the ceramics. So their so-called piezoelectric coefficients are often lower. But they have all the advantages that I would like for a mechanical energy harvesting device, namely, they're flexible, they're robust, they don't contain lead, which a lot of the ceramics, the common ceramic, piezoelectric ceramics do. So lots of things going for them. And so the question is, how do we make better piezoelectric polymers such that we can then use them in a device? So I've picked 4 candidate piezoelectric polymers. These are the ones we mostly work with in the lab. And as I go through, I'll talk about some of the kind of studies that we do with scanning probe microscopy, because the only way to really enhance piezoelectricity is to process these materials such that you can manipulate their structure at the nanoscale. Because think about it, ceramics, as I said, piezoelectricity is rooted in the crystalline structure. Ceramics, yes, well-defined crystal structure. Polymers, not so much. A lot of these are semi-crystalline, and even then, if you think of a polymer, you think of, a mesh of molecules. Even though you might have an individual dipole within the chain, overall, when you have this tangled, mass of chains, the dipoles basically all cancel out. So that's really where the challenge lies. So let's start with PVDF, or polyvinylidene fluoride, which is kind of a poster child for piezoelectric polymers. So as I mentioned, size matters, because when you go to smaller scales, you can control better the crystalline properties, and you can also control better the alignment of these chains. But so does phase. So phase is very, very important. And let me explain what I mean. So here's PVDF, the structure of PVDF. So you have a carbon backbone, and you've got hydrogen and fluorine atoms on either side. It usually crystallizes in the alpha phase, which is the thermodynamically stable phase. And you can kind of see that the hydrogen and fluorine atoms are sort of randomly distributed across the chain. Now, there is a dipole present in this structure. You have the more electronegative, you have a dipole going from the more electronegative fluorine to hydrogen, so it's perpendicular to the chain. But in this random configuration, you can kind of see that the dipoles basically cancel out. So this phase is not piezoelectric. In order to induce piezoelectric, I need a sustained dipole, right? I need a polarization in the chain. And for that, I need this beta phase, where you have all the hydrogen and fluorine atoms lined up nicely across the chain. So this is the all-trans configuration. Here, I do have a polarization, a net polarization in this structure. But remember, just having a lot of beta phase isn't enough, because at the end of the day, you want all of these, or as much of this phase, to also align once you have like when you have a lot of these chains together. So going back to what I said at the very beginning, we know that processing can be used to manipulate or to control phase and microstructure, and we do this using a simple template wetting method in the case of our piezoelectric polymers. So you start with a nanoporous template. So this is basically these are actually commercially available anodized aluminum oxide membranes, which are used for filtration. So they have sort of nano-sized pores. What we do is we either dissolve the polymer, or we melt it, and then we force it through the porous template. This could just be by capillary action, or by adding some sort of pressure. Once the polymer fills the pores, then we either do a controlled evaporation if it's from solution, or a controlled solidification if it's from the melt, until you're left with just the template with loads of these nanowires grown within it. You can see that they're all nicely aligned because they're grown within this nano-confined geometry. So it's quite a simple, scalable, versatile technique. And just to show you, so this is actually my first PhD student, Richard Whiter, and we showed that by growing these nanowires. So first off, there's some nice SEM images kind of showing you what these nanowires look like in the crack. You can actually get rid of the template. You can dissolve it away to reveal just the nanowires, or you can isolate single ones to do to do more measurements. But the first surprise, really, this was serendipity, is that if you look at the X-ray diffraction spectrum, so the blue line here shows the starting material, so the powder that we started with. And as you would expect, we have different crystallinities, alpha, beta, there are others as well. But when we made nanowires of this using this method, we seem to mostly be able to get our beta phase, which, like I said, is the phase that we're after. So we're already on the right track here. The other thing we observed is that you could make devices out of this straight away. So I was keen to see whether this works. And I was like, right, brute force method. Let's put some electrodes across this device. So here's our template with all the nanowires. We put some electrodes. So this is kind of what it looks like. So it's a very thin membrane packed with these aligned nanowires. And the first experiment was just tap on it, like just apply a little pressure and see whether you can see an electrical response. And you do actually get a current and voltage output. Of course, these experiments were done with a shaker. I didn't have the student tapping around all day. So this is a shaker where you can control like the amplitude with which you're applying pressure and the frequency as well. And it works out really well. What we found is that if you look at the energy conversion efficiency of this material here, it's about 11%. And to put that into perspective, if you do the same experiment with just a film of PVDF or bulk PVDF, that number is about 0.5%. So this is already quite a large improvement. But the most remarkable feature that we found is that these nanowires are self-poled. So let me explain what I mean by self-poling. So remember I said, ceramics, fantastic, they give you great piezoelectric properties. But even within a ceramic, like if you have a chunk of barium titanate, say, it's still made up of individual grains. And while each grain has a dipole moment associated or some polarization associated with it, if they're all pointing in random directions, same problem with a polymer. You can squeeze all you like, you're not going to get anything out of it. So what you need is for all the dipoles to point in a particular direction. So then when you apply pressure or strain or whatever, you change the overall polarization. In order to do this, you whack the material with a large electric field. It's called poling, electrical poling. You apply a large electric field, forces all the dipoles along or more or less along the direction of the electric field, and then you have yourself a piezoelectric material. Same rules should apply for the polymer. You should have to apply a large electric field. And in fact, for films, people need to do this, except for polymers, they often break down before you can even reach that sort of coercive field that you need in order to get this alignment. But what we found is we didn't need to pole. So in this device, these were as fabricated nanowires, and they were ready to go. So this there was something in this growth process, and we kind of understand a little bit of how that is happening, where essentially as the polymer is infiltrating these pores, they're getting stretched along the direction of the axis of the pore. And this is actually another way by which polymers are poled. They're like physically drawn in order to give them that alignment. We believe that's sort of what is happening here. And we discovered this in other piezoelectric systems as well. So this is this nice review paper that we published a couple of years ago, where we kind of show how the polarization is induced, usually either by drawing or by poling. You can see large electric fields are required. So this is the case in a case of PVDF, nylons, polylactic acid. This particular material is not ferroelectric, so you cannot actually pole it. So the only way to actually get inducement is by drawing. But we don't do any of this. Just the method of this nano-confined growth is enough. But we need to understand these properties at the nanoscale. And for that, we turn to good old AFM-based methods. Some of you might already be familiar with, in this case, we use piezo response force microscopy, where we use an AFM tip to apply a field across a sample. And if it's a piezoelectric sample, it will deform because you're applying a field across it. And you can measure that deformation with your AFM, the optical setup. So that's how it works. You apply an AC bias and see whether the surface of your sample is sort of getting displaced at that frequency. The problem is, as you can see, it's a sharp tip. And I'm talking about soft polymers. They don't really go. Also, I'm working with nanowires or individual nano objects. And and this is a contact mode method. You have to have contact. and you're sort of like scraping this tip across this soft sample. So you end up either displacing or destroying the material. So it's quite tricky doing PFM on polymers. So along with so this is Yartan was a postdoc working with me at the time. He's now faculty at the Technion. We came up with this new PFM technique for non-destructive characterization. I won't spend too much time on this, but just to say that sometimes you have to modify the experimental setups in order to make them work for you. But in this case, rather than scanning the tip continuously in contact mode, we use a sort of an intermittent contact method by which we can apply a very high frequency AC field just at the point of contact, and then we can sort of deconvolute the response to work out what the piezoelectric response is. So this is a very powerful technique, and it kind of opened up the door for us to be able to understand or study these materials better. So just a quick example of what these look like. So on the left, you see topography images of these nanowires, PVDF nanowires. And if you look at the right, these are PFM images. So this is kind of showing you the piezoelectric response. And you can see that even just as they are, you see that there is already a sizable piezoelectric effect. You can enhance that by actually using the tip to induce some polling. So you can apply a DC bias across what we did that around the middle to show that you can induce the polarization even further, but just to show that even without this, the material is already inherently self-poled. We try to look at other processing techniques and how this affects piezoelectricity. So this is some work we did with my good friend Ula in Poland, who's an expert in electrospinning. So this is a technique where you can make these sort of fibers, nano to micron size fibers of pretty much any polymer. And the way it works is you start with a solution of the polymer through a syringe, and then you apply a very high voltage, often of the order of tens of kilovolts, between the syringe and a collector. And the sort of the electrostatic repulsion, once it overcomes the surface tension at the tip, will let out this jet of the polymer, and you can use this collector then to collect this sort of mat of fibers. But there are quite a few things that you can vary. So it's a processing technique. So you can vary the temperature, the humidity, even the sign of the electric field. So whether you're applying a positive bias or a negative bias, all of these have an effect on the eventual properties of the material. And this is what we wanted to find out. So on the surface, if you just look at them, you probably can't tell much of a difference between, say, when you deposit at 30% or 60%. But when we start to use our AFM-based techniques to look at piezoelectric response, so again, at the top, here are a bunch of nanofiber or quite largish fibers that are produced under different conditions. So the numbers there represent the humidity, so that's 30%. The signs there represent whether we're applying a positive or a negative voltage. So they look the same-ish in topography, but when you start looking at the PFM images, you can start to see some differences. And that's kind of summarized in this table here. So this is looking at PFM amplitude for these different fibers that were produced under different conditions, and you can straight away see a winner. So it was the same starting solution, right? The PVDF you started off was the same, but then you end up with conditions that give you much higher responses for one. So this was really very useful, a really useful technique to try and understand how processing affects the properties. So I showed you an example for PVDF. Let's move on to some other less well-studied polymers. So polyamides or nylons also have a dipole in their structure, particularly if they are an odd-numbered polyamide. So by odd-numbered, I mean that there's an odd number of carbon atoms between the amide and the CO. That ensures that there is a dipole present in the system. If you have even numbers, then they sort of they cancel out. So if you start with an odd-numbered nylon, so in our case, we started with nylon 11. So this was work that was done by Anuja, who was a postdoc at the time. She's now faculty back in India. So she wanted to see whether we could use this nano-confined growth process to enhance piezoelectricity in nylon. So it's the same method, of infiltration, and then this work was then followed by, Yonsik was a PhD student in my group. He's now faculty back in Korea, in Yonsei University. He spent a lot of time trying to perfect or to understand the different stages. So, the point of infiltration, the nucleation, and subsequently the growth of this material within these pores, and how we could manipulate this using various external parameters like the temperature of infiltration or gas flow or the controlled solidification process. And all of this has been summarized in this nice review article here, so I won't go into detail. But the main point I want to make is that using these techniques, we can precisely control both the phase, which remember I said was important, and hence the piezoelectric properties in nylon 11. And just to say, nylons have been known to have piezoelectric properties. They were studied widely in the 80s and the 90s, but then their properties weren't as good as say PVDF, so they kind of died out. Also, they're hygroscopic, so not very stable. But we found that when you grow them in this technique, one, you can actually access phases that are not accessible in thin film or bulk form. For example, this so-called delta prime phase. So normally you have this is the thermodynamically stable alpha phase, where you have these chains that are very strongly hydrogen bonded. So there is a dipole present, but there's not much you can do with it. They're quite they're actually very strongly in place. You cannot change the polarization quite easily. So this doesn't really give you a very strong piezoelectric response. Whereas this delta prime phase, which we don't normally see, it's a metastable phase, this has a rotatable dipole moment. So we did some molecular simulations to work out, what phases would likely give us the largest polarization in the chain, and this is the one that we were after. So Jonsik spent a lot of time trying to get the right conditions in order to be able to make this delta prime phase. Again, you do not see this phase in the bulk or in the films, but only in nanowires. And then when we do some piezo response force microscopy, look at the PFM amplitude as a function of applied voltage. So the higher the voltage, the more the deformation. You can see that again, there's a clear winner here, and that's from our delta prime phase. So that's another nice example of how you can control piezoelectricity in these structures. But around the same time, we came across this phenomenon, it was making quite a bit of buzz, and that's the triboelectric effect. So this is an effect that you are all familiar with. We've known about this for millennia now. And that's where you have charge transfer that takes place between two dissimilar materials when you rub. It's what we commonly, refer to as static. And often you want to get rid of static, right? You don't want that because it can destroy electrical components. But it is a way by which you can actually convert sort of the friction between two materials into charge that is generated and then that you can harvest and use in a circuit. So for this to work, ideally, you want to pick two materials which are, one which is very likely to give up electrons, one which is likely to accept electrons. So there are these empirical triboelectric series that allows you to choose. So you've got more tribonegative to more tribopositive materials. You can see lots of synthetic polymers down here in the tribonegative end. But on the tribopositive end, mostly things like skin and hair, which is no good to make devices, with one notable exception, and that happens to be nylon. And we were working with nylon. So we thought, right, let's see if we can make this into a triboelectric material. So in the case of a triboelectric material, the property that we're most interested in is the surface charge, right? The higher the surface potential, the more the surface charge, the more likely it is to generate this or to transfer this during a rubbing event. So in this case, we use what's called Kelvin probe force microscopy, which allows us to map the surface potential of these materials. So here are two examples. So the top images are all topography. And the bottom images are all sort of looking at the surface potential at the top of these nylon nanowires. And in the first image, we just sort of measure what the surface potential is. In the second image, we actually zoom into a smaller subsection of the image, and we use the AFM tip itself as the rubbing element, right? So we use that to transfer some charge, and then you could zoom out and kind of see how much charge you managed to transfer. And you can clearly see that there's a difference on the left to the right, and the main difference is that on the left, we have what we produced as delta prime nanowires, and on the right is the alpha nanowires. And you can see that the alpha nanowires have a much larger transfer of triboelectric charge than the delta prime. So just to recap, remember, the delta prime was great for a piezoelectric, from the piezoelectric point of view, but it looks like the alpha nanowires are actually much better for triboelectric applications. Again, the only way to see if this is true is to make a device. So a typical device would have two different parts. So you've got your nanowires on one side. We use Teflon, which is very tribonegative, on the other side as a counter material. And you've got electrodes on either side. So as you tap or rub, there's some charge transfer that takes place. Charges are induced on the electrodes, and then it'll flow through an external circuit. So this is kind of what it looks like. We've done this using aluminum instead of the nanowires, and then our delta prime nanowires, alpha nanowires. And what we're looking at is the current density that you get as you excite this device. And if you look at the power density, so at the end of the day, we want this to do some work. So if you have a bunch of resistors across which you're sort of trying to dissipate the power, then the highest power output you get is for the alpha nanowires. And this was surprising to us because, we thought, well, the alpha nanowires were not of much use from a piezoelectric point of view, but they work really well, and in particular in the nanowire form, because what they have is they do have a dipole, and this dipole is very strongly bound due to the strong hydrogen bonding between the sheets. And because we have this alignment within the nanowires, it's always pointing in the right direction. So you don't see this in films, because in films, again, you have different lamellae. Each lamellae will have a dipole moment, but they're pointing in different directions. So net effect is nothing. So this only works or manifests in these nanostructured samples of nylon, which is, and they're very thermally stable. So you can heat this material, bring it back down, and nothing changes. It's still able to hold on to that polarization. So we extended this further. This was Tommaso, who's a student in my group. What he tried to do was to see whether you could make fibers, right? With nylon, you think, okay, maybe this can be textiles. We're constantly moving. As fibers rub against each other, maybe we can produce, some usable electricity for small power devices. So what he did was he started to electrospin PVDF and nylon onto conducting cores. So we need an electrode at some point, right? Because we need to collect the charge. So the core he uses is a carbon nanotube yarn. And he then basically electrospun PVDF. You can kind of see what it looks like in cross section. So that's our electrode essentially, and this is the PVDF. And it's great because you have a lot of surface area, because these are fibers, which is also what you want for a triboelectric device. The more the surface area, the higher the surface charge. And these are the kind of fibers. So we call them textiles, but not quite there. But again, you can use things like KPFM. In this case, previously we used PFM to optimize condition. Here we're using Kelvin probe force microscopy to optimize condition. So these are basically different fibers which are grown under different humidity conditions. And you can see that one gives you a much higher surface potential. So that's the one we want for our triboelectric devices. And finally, you can actually make textiles out of this. So here we've combined a tribopositive, this should actually be the other way around. It's a tribopositive nylon 11 yarn and a tribonegative PVDF yarn. You weave them together. I say you weave them together. Really, that's our textile. This is done in the lab, but hopefully can be upscaled. And this is the idea. As these two rub against each other, they do produce a sizable power against a load resistance. You could use it to charge up capacitors and things like that. We showed that it was washable. So apparently for something to qualify as a textile, you have to show that it is washable. So we stuck this into the washing machine a few times, and it works. So that's work we've done with nylon. I'll move on to the next one, which I want to talk about, which is polyl-lactic acid, or PLLA. Again, some of you might already be familiar with this. This is used quite often as scaffolds for wound dressing. So we know that it's already biocompatible. It also happens to be piezoelectric, but it has a strange kind of piezoelectricity called shear piezoelectricity, because it's you can see it has a chirality, and the dipole sort of goes around the chain. And what this means is, rather than squeezing the material or stretching it to get an electric response, you can bend it, like you can shear the material in order to get an electric response. And I'll show you where this is quite useful. So again, we do the similar treatment of growing these nanowires, again, because we want to get the alignment of the dipole, we want to control the crystallinity. The reason the shear effect is so interesting is because what the idea was to create a cell culture device, actually, where we can use the piezoelectric effect to both apply a force and also to detect what is going on. Because remember, I can have an array of these nanowires. I can apply a field across them, and it'll cause them to bend or to stiffen up. And if there's anything that's happening at the top which is causing these nanowires to bend, I should be able to detect this as an electrical response. So sort of using this as an interface with cells. My student Callie did quite a bit of work on how to control the crystallinity. So in her case, she was controlling the crystallinity of these nanowires using as a function of the annealing time, as you can see. So she did a lot of X-ray work and worked out what the ideal conditions were to get highly crystalline nanowires. So again, starting material is the same. You can go from almost amorphous to in this case 40% crystalline. And Mike, who was a PhD in the group, he then started these first experiments where we started to put cells onto these nanowire arrays just to see what they do. And the first thing we noticed is they loved these nanowire arrays because these are, basically rough nanostructured surfaces. So you can see these are human fibroblasts, dermal fibroblasts, so skin cells. They let out these cellular projections. They're grabbing, they're pulling, which is exactly what we want because this means that they're shearing these nanowires and we can detect this. And he did this for amorphous as well as crystalline nanowires, and we saw a difference in the way the cells behave. So here we look at cell attachment for films of the material. They don't like the films at all, so almost 0% attachment. And here are the nanowires. You can see they attach more strongly to the crystalline ones than to the amorphous ones. And TCP is just tissue culture plastics. This is what people normally use. So this is as a control experiment. So we're like, right, the cells didn't do any X-ray diffraction, but somehow they worked out which was crystalline and which was amorphous. So what exactly are they responding to? So what they're responding to is a variety of, it's two main things. One is the stiffness of the material. So with the crystalline nanotubes, one would expect should be stiffer. But we use this method called quantitative nanomechanical mapping. It's again an AFM-based method by which you use the tip to deform the material to see how stiff it is. So the further you deform, the softer it is, right? So here are amorphous nanotubes, here are crystalline nanotubes, and you can see that the amorphous ones are more easily deformed, which means that the crystalline ones are stiffer. We know that cells respond to the mechanical environment, to the cues that they get from the, for instance, the stiffness of the substrate that they're on. So we believe that this is playing a part. But that's not all. There's also a difference in the surface potential. So there is an electrical effect going on as well, where the crystalline nanowires have a slightly elevated surface potential. And finally, if you look at the piezoelectric response, here too, the crystalline nanowires have a higher piezoelectric response. So we believe it's a combination. It's actually the electromechanical environment that is having an effect on cell behavior. And we can harness this because what you can do is you can use nanostructuring as a tool to tune the effective stiffness. So rather than, if you look at the body, you've got all ranges of stiffnesses starting from brain all the way to bone, whereas most of the experiments we do are in tissue culture plastic with one set stiffness. And even if we were to replace these with piezoelectric materials, these are still at the stiffer end. However, you can change the stiffness. You can manipulate. This is a nice engineering trick by nanostructuring. So if you have structures with long enough aspect ratios, you can see that the effective stiffness actually goes as the inverse square of the aspect ratio. So basically, the longer and more slender they are, the bendier they are. So we can use this therefore to kind of to tune the mechanical environment for the same material, keeping everything else the same, crystallinity, surface potential, etc., to see what effect this might have. So my student Thomas Chalkman did this using iPSC-derived endothelial cells. So we did this work with the Cambridge Blood Center, where they were trying to improve the amount of endothelial cells they were producing from their normal chemical concoction. And this is an example of what they have. So this is looking over 14-day incubation period of iPSCs. So this is the standard well plus vitronectin. You put down your cells, you start the differentiation process, and this is what they end up with. Whereas the same experiment, when we do it on our chips, you can see this is about a 67% improvement in the yield. So there is something about being able to control that mechanical environment, and possibly because these are piezoelectric polymers, the electrical environment that is giving rise to this effect. So that brings me to the last polymer I want to talk about. So this is collagen, which is a naturally occurring polymer. This is work which was done with Malavika, who was a research fellow in my lab. She's now assistant professor in Oxford. And she was she came up with this idea of trying to see whether we could induce piezoelectricity in collagen bundles. So collagen again is a molecule where there is a dipole, but again, because there is no overall alignment, I mean, when you don't have any overall alignment, you don't see any net effect. But if we can induce localized piezoelectricity in collagen, then this could be used towards cell growth, tissue regeneration, etc., which is what Malavika is currently focusing on. But in my group, we tried to do this experiment where we looked at sort of non-cross-linked collagen versus cross-linked collagen using different cross-linkers. So these are different cross-linkers that you use. And the first thing that she saw is one, the stiffnesses can be quite different depending on the cross-linking that you use. But more interestingly, I think at least, is that there's a difference in the piezoelectric response. So here are some, so that's topography, and the rest of those basically are piezoelectric PFM images. So this is for non-cross-linked versus cross-linked collagen. And if you look at the lateral amplitude, because this is again another molecule that has sheer piezoelectricity, so you sort of see it in plane in this case, you can start to see the emergence of bundles in these cross-linked samples, which also have a piezoelectric response attached to it. So if we can control this, then you can have, you can have a device where you have certain sections where you have controlled induced piezoelectricity that you can address electrically from the outside. So this brings me on to another very interesting technique that we use, namely aerosol-jet printing, which is a wide area fine feature printing technique that you can use to print anything from metals to polymers to anything that you can make an ink out of. You could pretty much use this to print. The main advantage that this has is that you can go down to very small the resolution can be quite small, so down to 10 microns. So anything that you would do with photolithography, you can do by direct printing with this. And it's a technique we use to make devices quite easily. This too is a processing technique with many parameters that you can control. For instance, I won't go into details, but there's all sorts of flow rates and sheath flow rates, etc., which you can use to change the morphology and hence the properties of the material. So what Malavika was trying to do was to see whether we could actually print collagen inks to see whether you could induce piezoelectricity just using the printing process itself. And so there are loads of images here. I don't know how clearly you can see this, but depending on the conditions, so she varied what's called sheath flow rate, atomizer flow rate, etc. You can actually see there are, under certain conditions, there's this bundling effect that happens of these collagen fibrils. And this is very akin to what we have in muscles, actually, where there are regions where you have aligned collagen fibers, and that's where you have this piezoelectric response manifest. So she was able to do this kind of synthetically in the lab using this printing process. I don't know how clearly you can see them, but there are these fibers, which then if you, for instance, now use KPFM to look at surface potential, to look at the PFM amplitude, both in phase and out of phase. Again, quite a lot of data to decipher. So what I can tell you is that the lateral piezoelectric amplitude, so the shear piezoelectricity, using our printing technique is about 2 to 4 times larger than just simply drop casting these collagen samples. And this is consistent, in fact, with the alignment-driven increases that you notice in things like membranes and tendons, etc. So it's a way of replicating what we find within the human body. So I just wanted to put this up here to say we've done a lot of work over the years on all sorts of piezoelectric and triboelectric polymers, mostly from the point of view of energy harvesting. As you can see, the latest one actually just came out last week. So there's plenty of scope really for these materials in this area. And if anyone's interested, I'm really happy to have a chat. But I wanted to pick up on this technique because I think it's a really versatile technique, and we've had so much fun with this over the years. So I'm just going to spend the last few slides just telling you a little bit about some of the other things that we've been doing. What this does basically is if you have an ink, it atomizes it, makes these droplets, fine droplets out of this ink, and it jets it out through a sharp nozzle. Now, it's kind of like a spray can, right? But if you have a spray can, you have droplets going everywhere. So in order to focus it, there's the sheath gas, which I was talking about a little while ago, which then focuses it down. And then you can make circuits, like I said, down from 10 microns to sort of large scale structures. And it's a rapid prototyping technique, right? Like within an afternoon, you can try different geometries, different samples, and you can layer material. So it's not just one type of material. You can actually build circuits with this. So one of the things we showed actually is that you can, because it's like essentially spray cans, and we have 2 atomizers, you can have in situ mixing of organic and inorganic components. So you can imagine that you've got maybe a polymer or some organic component and a nanoparticle dispersion. And you can have very good mixing while they're in the aerosolized form, such that you have near ideal dispersion without needing, for example, surfactants. This is particularly important if you're working with, maybe some electronic components where having surfactants or having interfaces or just having clumping of nanomaterials can be a problem. We see none of that because this, like I said, this mixing doesn't happen sort of, you're not like physically mixing or anything. This is actually happening in spray. And we earlier published some work showing how you can get things like, high K dielectrics with using this technique. But also my student, Canlin, actually showed that you could mix, for instance, an organic conductor. A lot of you here work with will probably recognize P.PSS. And an inorganic component. So in this case, we use bismuth telluride or antimony telluride, which we've grown ourselves using solvothermal synthesis. We make an ink, and we sort of we deposit these two together to make a composite. And what Canlin was doing at the time was looking at the thermoelectric properties of these composites. And what he found was that, again, by changing the loading ratio, by changing the amount of the inorganic versus the organic component, you can start to find compositions where you have sort of the largest power factor, which is what people working in thermoelectrics are interested in. And again, it's also possible to induce compositional grading. So rather than have one material with one composition, you can actually spatially vary the composition, which for thermoelectrics is actually quite useful, because rather than having one material that has a fixed ZT at one temperature, you can actually design the material to have, to maximize the ZT as you go along the temperature gradient. So there's some nice work which Canlin had done. Just to kind of show you some other cool things we do, you can use these to also produce 3D structures. So we're doing some work on 3D metamaterials. This is work we did with Jenny Zhang from the chemistry department in Cambridge, who was interested in this bacteria called cyanobacteria. So these are photoactive bacteria. You shine light on them and they produce electrons, right? So you want to wire them up, right? We have to wire them in order to be able to extract that. And typically the way people would do this is, you have a flat conducting substrate and you put your cyanobacteria on top of it. Yeah, you get some photocurrent out of it, not very high, because at the end of the day, you want more coverage of the bacteria. So what's the next thing you can do? You can make them nanoporous or microporous, basically increase the surface area so that you have more bacteria attaching to your electrodes. What we showed was actually using with aerosol-jet printing, we could actually make these 3D structures. And by playing around with the processing parameters, you could include porosity. So it was sort of like a hierarchical 3D micropillar. And now, and these are ITO, by the way, I should have said that. So these are indium tin oxide because you need the material to be transparent for the light to get in. And now, once you coat these with the bacteria, shine the light, it's party time. You can see this was, I think, the highest photocurrent that was ever reported for this sort of system. Another very recent work, so this is actually about to come out very soon, is work we did with Jin Chong in Oxford, where he works with MOFs, and he uses these MOFs as hosts for luminescent dyes. And what we showed is that you could actually print them. So these are actually prints of these various morph structures which are infused with these dyes. And when you look at this under UV, you can see the luminescence. You can do this with various dyes, so yellow, red, green, whatever. And then you can mix them all in different proportions to the point to see different colors. We've even shown in this paper that you can get white light emission from this. So it's a really versatile technique, and you can do this sort of in almost like a smooth organic process, rather than mixing and trying and seeing it. You can actually do this variation in the lab over a day. So I spoke a lot about energy, and just the last few minutes, I think I'm just going to quickly show you some stuff that we do across sensors and actuators for biomedical applications across length scales. So when I say across length scales, I mean sensors that can be used for hip replacement surgery. So this is work done by my student Liam and postdoc Chin Cheng, who's now faculty in Glasgow, where we actually used a microfluidic device to build a force sensor. And the way we did that, and again, this is thanks to aerosol-jet printing, is we were able to print directly within a microfluidic channel an electrical circuit. In our case, it's a simple capacitive circuit. So this is a channel, there's some interdigitated electrodes. As you apply force to the reservoir here, it pushes the liquid up and down the channel, and that changes basically the electrical properties or the capacitance of these electrodes. And this is what we measure. And it's a nice linear dependence. So the more you push, The more fluid gets displaced, the higher the change in capacitance. And this can be, nice and conformable. And the main advantage is that you can measure quite high forces by playing around with the material. So if you have a stiffer, a stiffer channel material, then you have to apply higher force to move the liquid. And this is all important. Why did we do this? Is because we wanted to be able to measure the forces that go through the hip joint during hip replacement surgery. So I didn't know this, but apparently, not apparently, this is true. When surgeons replace hips, when they go to put in the final implant, the positioning is done by feel. Right, so they go through the motions and they think, okay, I think that feels about right, and then they go for it. And sometimes they get it right. If you have a very experienced surgeon, then your chances of this working are great. But if not, you could be in trouble. In fact, the surgeon I work with, he runs what's called the young adult hip clinic. So he sees patients in his 20s, in their 20s, 30s, 40s, and they want to have an implant that lasts a much longer time. And also they want to do, stuff which young people would want to do. So getting this positioning right was very, very important. So they need to be able to measure the forces for an objective balancing. But they couldn't do this because there were no sensors that could A, fit into this tight joint, and B, measure the large forces that go through the joint. And that's what we came up with this. And this is sort of what it looks like when we put them into the implant. So that's our array of sensors, and it goes in to the implant. And now when the surgeon is positioning it, they have the full readout from these sensors. Mind you, we don't leave these in the body. They're only used during the trialing phase, which is what they normally do. And then once you get the right position, you chuck it out and you put in your final sensor. So this actually led out to a spin-out company, Artiosense, which is now doing quite well. We just got quite a bit of funding from the National Institute of Health Research back in the UK. And so we are moving towards our first in human trials, hopefully in 2 years. And then on the other scale, so this was, sensing big forces. This is, again, very recent work. It just came out, I think, a couple of months ago, work done by my students, Diane and Tom, where we are using the same platform, so a microfluidic channel with these interdigitated electrodes. In this case, we're just looking at the impedance or effective capacitance, if you will, as a function of frequency for different concentrations. In this case, the example shown here is sodium. And what we notice is that there is a turning point that you can measure from these capacitance versus frequency curves. And if you plot those, so this is sodium, they actually scale linearly with the species concentration. And this works for sodium, potassium, we've done it for a bunch of others. So it's a very there's no chemistry involved. There's nothing. We're just looking at the electrical response of these liquids. So this is basically what's going into the detection of electrolytes and eventually metabolites. Of course, for metabolites, you need something that can identify the metabolite that you're after. So currently, the work that's going on in my lab involves actually making ion selective membranes and molecularly imprinted polymers for improved selectivity. So that's quite exciting. And finally, we also do some actuation. So there's sensing, but now there's actuation again for soft robotics. So Kalyan, who's a Newton International Fellow in my group, he's been working on these shape memory polymers, and G works on printed tri-layer electrochemical actuators. So just really very quickly, what Kalyan does is he makes his own resins, and by changing, for instance, the amount of cross-linker that he uses, he can change the glass transition temperature of these shape memory polymers so that they work close to room temperature. So these wires that you see, actually each of these fingers has a heater in it. So you can pass a current, heat it up, and then it deforms. So yeah, so we have quite a lot of fun with these, as you can imagine, in the lab. And on the other hand, what G has been doing is he's printed this tri-layer electrochemical actuator, which basically consists of P.PSS on either side, sandwiching Naphion here. And basically, when you apply an electric field, you have oxidation reduction on one end. You have these protons which move across, and that causes this bending. This entire structure is aerosol-jet printed, and so this what we reported here is, I believe, the thinnest electrochemical actuator. It's about 10 microns, very, very thin. And also we can pattern it. So here you can see there are actually two segments. So we can, rather than bend the whole thing all at once, we can actually control individual segments. So you can get all these funny movements. So why did we do this? Because we also work with someone who inserts cochlear implants for hearing impaired patients. And this has to be done really, really carefully, because if they bump into the basal membrane, for example, while inserting, then you could end up causing more damage, getting rid of any residual hearing the patient might have. So they came to us with a problem like, can you have some sort of guided insertion where, you can have a system like this, which can sense when it's getting close to a maybe the basal membrane, say, and can kind of self-correct and move away. So that's basically what we're developing this for. So in this case, it has to be like really, really thin, really small, and that's why we turn to printing. So that's me. I've hopefully touched upon all of these in little bits and pieces. I need to finish by, of course, thanking my fantastic group. I wouldn't be here without all the hard work that they put in. So members, past and present. You can find out more about us on the website. I'm also on Twitter. And of course, thanks to the funding agencies. Oh, yes, one last thing, if I may. So this year I've been appointed as the inaugural editor-in-chief of a new journal called APL Electronic Devices from AIP. And we're now open for submission as of, I think, just a few days. So very excited. I just thought I'd put that out there because I know a lot of people here might be interested. We cover a lot of different, it's quite wide in scope. But if you're interested, please do have a look. And we'd love to hear from you. But yep, that's it. That's me. Thank you very much.
Thanks so much, Sohini, for the wonderful talk. I'm sure there's some questions, so please just go right ahead.
Thank you for the talk. So I was wondering about the directionality of these piezoelectric effects, because you were showing initially these fibers that you grow in the membranes.
Yes.
And then you put the electrodes across the membrane, but then the AFM you're doing was sensing sort of the walls of the fiber. So if you could comment on how the directionality matters, how do you polymer orients?
Yeah, so that's a great question. So when I say alignment, what we have actually is more texturing along the direction, along the long axis of the pores, right? So because that is the dominant direction along which, one, you have this sort of drawing effect, but two, the way these materials grow is you have nucleation along the walls, and then those grow, and that flow allows more or less alignment along the axis of the pores. But that is not to say that it is a perfect alignment, right? So you will still see the effect from the sides. And yes, you're absolutely right. When we do the when we're looking at the self-polling on the nanowires, yes, we do have them lying flat, but you can also see that when you apply an electric field, you're able to also rotate the dipoles to point in the direction of the electric field. So yes, it's and the other way that we look at it is when we actually look at KPFM data from the tips of the nanowire, and there you do see that there's an enhanced surface polarization. And the reason for that is because, again, you have this overall texturing which is along the wire. So it's not perfect. So it still works. So we say it's self-polling, but it's it's not like all the dipoles are pointing. We would never get that in a polymeric system. So we would always expect there to be some response from the other sides as well. But it's diminished compared to what we would see along the axis.
Thank you. It has been so many different things.
Yes.
I have many questions, but the last starting at the end, when you use this finger with PDOT, you have shown the mechanism with an interface of Nafion.
Yeah.
So that is proton, the one that is moving from the cathode to the anode.
Yes.
But if that was to be placed in the arm of somebody, will it be it will be wet with acid, or it will be? I mean, it has been done before with gold and with sodium intercalation. I don't know.
Yes. So these are all in air, and we hydrate the Nafion membranes. So it can operate within, like, in ambient atmosphere. But yeah, if once all the moisture is gone, then it stops to.
Oh, it will be just enough with moisture. You mean?
So
okay.
the kind of the application that we have in mind is, it needs to work for the amount of time, for instance, that we are inserting, say, some implant, right? So it's not staying there and actuating forever. If that's
it's not going to stay inside the body. It's outside. It's a robot.
Exactly.
It's like the hand.
Yes, exactly. Yes. So it's only meant for the amount of time that we need it to work during and then insertion. But it's not once it's in there, it's not doing anything. It's for guided insertion. But you're absolutely right. Like once you're out of moisture, it's not going to work.
I also had another common question, like it seems like cells like fibers,
Yes.
like collagen or other things.
Yep.
But there were some studies at some point that if you make like two fibers crossing, they don't like the 90-degree angles,
Okay.
but they like the 120 degrees.
Okay.
I was wondering if you have checked the angle of between the fibers or.
That's a good point, and I don't think we have, because I'll see if I can find the image. In our case, so this is sort of looking at it from the top down.
These are from the top.
Yeah, so these are all just, fibers, and imagine the cells which are, but no, we haven't done a controlled experiment like that, but that would be quite interesting. I mean, of course, we see some, as you can see, we can see some clumping. Yeah. And this is partly because of the way the cells are kind of tugging on these, but no, I haven't actually seen. I guess for that, what we would need to do is to lay these flat.
Yes, it was flat. It was a flat
yeah.
thing. It was
no, and then that is possible. So yeah, worth looking at.
Just not to ask too many.
No, please.
My last curiosity is, do you think any of these can be applied to sensor the glucose?
Oh, yes. So
you will get very rich.
So
if you don't.
That's a good question. So when we started to work on these wearable sensors, we actually got some money from a company, but because it's a company, the first thing they wanted to know was go and do some market research, like what are the things people want to measure, and glucose was obviously at the top of it. I don't have the data to show here, but this can actually be used for glucose monitoring. It has a completely different response to what you see here, because this is due to the fact that you have ions, you have these electric double layers that are forming at these interfaces, whereas with glucose you have none of that. So you do see a response, but it's not one that I can quantify in this way. However, we started working on, so I have it here. We started working on molecularly imprinted polymers because one of the other metabolites people were interested in was lactate. And we used a boronic acid-based polymer for detection of lactate. And it turns out it detects glucose as well, right? And the response is different. So we have actually started to, yes, but the trick there for us, the main challenge is to control how much of the molecularly imprinted polymer is there, because that's the only way we can quantify what is attaching to it. And that's kind of our bottleneck at the moment. But yes, absolutely. Glucose is high up there. And the nice thing is, here we don't have any reactants per se. So, in principle, I mean, the detection mechanism is purely electrical. So it's quite different from, the normal electrochemical sensors that are out there. Thank you.
Okay. I've got plenty of time to ask you questions, so I won't ask them now. I'll ask them later. Last chance? Okay, if not. So I think it's been a great start to our seminar series for this season. So let's thank Sohini again very much.
Thank you very much. Thanks for having me.